Hello, in this video you're going to learn about Hox and Hox genes and how they provide evolutionary evidence for a link between species. So what are Hox genes? They are clusters of genes in the DNA or other animals, including mice. These genes are arranged in the same way from head to tail. The function of the Hox genes in other animals seems to be almost the same as in fruit flies. That tells that the cells of the body how to differentiate as they grow. So these genes are sort of like clusters of genes that tell your cells how to grow, and they're clustered from head to tail. And what's neat is that those genes are clustered in the same way fruit flies, mice, humans. So in fruit flies, these genes are in the same sequence from head to tail in a fruit fly as they are from head to tail in a mouse, which seems to show that there is an evolutionary link between the two. So we're going to look at that in more detail in this activity. Let's get started.
So I will be jumping in and out of the video you'll be watching to explain different things.
3. The evolution of Hox genes. Homeotic Hox genes were first discovered in fruit flies through the striking phenotypes that they give rise to when the genes are mutated. In these homeotic phenotypes, one part of the body along the anterior to posterior, or head to tail, axis is converted to the likeness of another part of the body.
So when they say anterior, they're talking or referring to the front of the body. So in a mouse, that would be the head. When they say posterior, they're referring to the back end of the body or the tail. Same thing for a fly. Anterior means the head, and posterior would refer to the back end of the body or the tail.
In this example, you can see the normal structure of the antenna on the head of a wild-type fruit fly here on the left. But in this fly on the right, that has a homeotic mutation, you can see that this antenna on the head has now been transformed into a leg structure, a structure usually only found on the thorax of the fly.
This is a better picture of the homeotic mutation. We have a normal fruit fly, and this is its antenna. And in this mutation, the legs have been transposed, and they are where the antenna should be. That's due to a Hox gene mutation.
Striking homeotic phenotypes like this are part of why the Hox genes have garnered so much interest. You can see in this diagram that different regions of the fly express different Hox genes. And it turns out that the normal role of the Hox genes is to provide regional identity to cells along the anterior to posterior axis of the body, so that cells have the information needed to form the correct structures in the correct places. Hox genes achieve this function by regulating other genes. The encoded Hox proteins can bind to DNA and regulate entire sets of other genes that are needed to make the correct structures for a particular region of the body.
That's a lot of information she just gave, so let's break that down a little bit. So first off, Hox genes follow an anterior to posterior body axis, which means that here's the order of the Hox genes on the DNA. And what happens is the ones that are further up on the DNA, they control this segment. The ones right behind that control the segment right behind it. The ones right behind that control the segment behind that. So it follows an anterior to posterior body axis, starting from the beginning and going to the end, or starting at the front and going to the back. And they are in the same order on the DNA. So how do Hox genes work? Well, in general, Hox genes work. Let's get my pointer. They control or regulate other genes which control functions. So Hox genes are working in an anterior to posterior pattern. They can control the formation of organs, organgenesis. They can control cell migration. They control cell growth. They can control or regulate differentiation, the cell cycle. They can regulate cellular identity and cellular interactions. So how do they do this? Well, Hox genes will make Hox gene will be on the DNA. The DNA will copy itself into a piece of RNA. That RNA then will go out to a ribosome in the cell, and that ribosome will take the directions or the recipe that was on this RNA and make a protein. Right? And that's how transcription and translation work. There are recipes in your DNA, they get copied, that copy goes to an RNA, which is basically a kitchen. The ribosomes are basically kitchens, and the kitchen then will make that recipe into a protein. That protein, if it's a Hox protein, that protein will then go and activate genes on your DNA so that they can be copied and they can form different parts of your body. Right? So they regulate what genes are turned on and what structures are made from that. So Hox genes make proteins that regulate other genes. So what does that mean? Well, if you have your Hox group here, this Hox gene would go to DNA and turn on the part that would form your scapula. Well, your scapula is right back here. Right below that, this Hox set will turn on parts of your DNA that will form your humerus. And your humerus is this upper part of your arm. The Hox proteins below that turn on parts of your DNA that form your ulna and radius. And the ones below that turn on parts of your DNA that form your metacarpals and then your digits. So the Hox genes regulate other genes that then will form body parts.
When the first Hox genes were sequenced, it became clear that they all contained a short stretch of 180 base pairs of sequence that showed similarity to all the other Hox genes. This stretch of sequence was called the homeobox, and it encodes the part of the Hox protein that directly binds to DNA. This stretch of the protein is called a homeodomain. So the DNA sequence is a homeobox, and the corresponding encoded protein domain is the homeodomain. The homeodomain generally forms a helix, loop, helix, turn, helix structure, and this allows it to bind to DNA. There are other genes, not just the Hox genes, that contain homeoboxes. Homeobox-containing genes, in fact, form a large and diverse family of genes, of which the homeotic Hox genes form just a specific subset. Excitingly, since their discovery in fruit flies, these Hox genes have been shown to play a role in patterning the anterior to posterior axis of most animals, including humans. Hox genes tend to be found in clusters in the genomes of animals, and changes to the exact number, sequence, and regulation of the Hox genes have clearly played an important role in the evolution of different body plans in animals, as we describe in Chapter 3. We can therefore make important insights into the evolution of animal body plans by tracing the evolution of Hox genes over time. We can search genome sequences for similarities to the Hox homeobox sequences as a way of identifying Hox genes in the genomes of animals that are around today. But how do we look back across evolutionary time at the history of these genes? Well, we can trace the evolution of gene families like the Hox genes by mapping the genes onto an evolutionary tree, a phylogenetic tree, of the species of interest. For example, let's take a look at Hox genes in vertebrates like ourselves. When we look at humans, we find Hox genes clustered together in 4 different chromosomal locations in our genome. Each cluster of Hox genes contains varying numbers of genes, indicating there have been duplications and losses of different genes in different clusters. And, based on sequence similarities, the Hox genes can be classified into 4 main types, indicated by the different colors here. But how did we end up with this particular configuration of Hox genes? And, more specifically, what did the Hox genes of the last common ancestor of all vertebrates look like? Let's take a look at an evolutionary tree of vertebrates. Those are animals with backbones like ours. When we look at the Hox genes of other mammals, like mice, we find that they have a very similar number of Hox clusters and genes within the clusters to humans. If we look across the tree a little bit further to birds and other tetrapods, those are the 4-limbed vertebrates, we also see 4 Hox clusters, but occasional loss and gain of some of the individual genes. When we look at even more distantly related vertebrates, like fish, we see more variety in Hox composition. The model organism zebrafish, for example, has 7 Hox clusters. Many other fish have 8 clusters, and salmon have even more clusters. And throughout the fish lineages, we also see variation in the composite of genes present in each of the clusters. So clearly there have been changes through duplications and deletions of individual genes, and duplications and deletions of entire clusters of genes, and even entire genomes over the course of vertebrate evolution. And we can see that to look further back in evolutionary time, to try to understand what the Hox genes of the last common ancestor of all vertebrates look like, it would be really helpful to reach further afield to more distantly related animals that are close relatives to vertebrates but are not themselves vertebrates. Animals that sit at this key position on the tree of life are non-vertebrate chordates, like this animal, Amphioxus. Chordates are animals that have a structure called a notochord. The notochord runs along the length of the spinal cord and provides structural support to the body. In vertebrates, the notochord becomes reduced as animals develop and as the bones of the vertebral column form. Those bones are the vertebrae that we and all vertebrates have in our backs. But in non-vertebrate chordates like Amphioxus, the notochord persists, and these animals don't form the bones of a vertebral column. These non-vertebrate chordates are the closest relatives of the vertebrates. Analysis of their genomes is therefore particularly interesting because of the potential this analysis has to shed light on what the genome of the very first vertebrate ancestors might have looked like. Researchers have therefore analyzed the genome of animals like Amphioxus to identify the Hox genes present. They have found that Amphioxus contains a single Hox cluster with 15 genes. It seems they've actually duplicated some of the more posterior Hox genes, giving them some extra genes in this group. From this, we can infer that the ancestral precursor to all vertebrates likely had a single Hox cluster of about 14 genes. This cluster then became duplicated twice, probably through 2 rounds of whole genome duplication, resulting in 4 Hox clusters in the last common ancestor of all vertebrates.
Okay, let's see if I can clarify the Hox gene duplication through evolutionary time a little bit. So we'd have our one Hox gene from our inferred ancestor, and if that's duplicated, then you would end up with this. Well, sometimes the duplication takes place where you end up with 2 genomes stuck together. So in one critter there'd be nothing, in this one there'd be 2. They were linked. Okay, so that would be a whole genome duplication where they're stuck together. And if these 2 then are duplicated and they get stuck together, you would end up with this. So we started off with 1, that 1 duplicated and stuck together, that left with 2, and then your 4. So that'd be 2 whole genome duplications, 1, 2, and then this one's double to 4, where you would end up going from 1 to 4 Hox gene clusters. And like if you keep on going with that, we could end up with the fish, and the fish had up to 8. So you have your 4 genome cluster, and if that's duplicated, you would get this. But if the duplication ends up with being a double duplication, you'd end up with 8 of them, right? Or in some fish, they don't have 8, they have 7. That's because you had a deletion. So you'd have 1, you'd have the double duplication, but one of the last clusters got deleted, so instead of having 8, you would have 7. So through evolutionary time, you can see that through whole genome duplication, you can get more of these, and it would make evolutionary sense.
Individual Hox genes then underwent some additional changes in the lineages, giving rise to modern-day tetrapods like ourselves. And at the base of the fish lineage, it's likely there was an additional round of whole genome duplication, increasing the number of clusters from 4 to 8 Hox clusters in ancestral fish. This was then followed by a variety of additional single gene or single cluster duplications and losses in the various different fish lineages. In the zebrafish lineage, there appears to have been loss of 1 entire cluster, bringing the total number of clusters from 8 to 7. While in the salmon lineage, for example, there appears to have been a 4th round of whole genome duplication, followed by some gene losses. Overall, the fish show quite a lot of individual variations in the exact composition of Hox genes, against this general backdrop of whole genome duplication to 8 clusters in the ancestral fish species. We can extend this same approach of surveying the composition of Hox genes, not just in vertebrates and their nearest relatives, but to all multicellular animals and their nearest relatives, to ask what the composition of Hox genes might have been in the last common ancestor of all animals. Here we have a simplified evolutionary tree, a phylogenetic tree, of multicellular animals. There are two main branches to this part of the animal kingdom: the deuterostomes, like us humans, and the proteostomes, like fruit flies. These are all considered multicellular bilaterian animals, animals that at some point in their life cycle have a bilateral or left-right symmetry to their body plan.
Let's see if I can explain the bilateral symmetry a little bit. So, bilateral symmetry just means that the left and the right side are similar, or they look the same. So in fruit flies, humans, and higher animals, or more evolved animals, they have bilateral symmetry. In starfish, they do not have bilateral symmetry because there are more than, there's not just a left, right, the same. They're round. Okay, in conjunction with this, if we go back even further, we have sponges, and sponges do not have any symmetry at all. We have starfish and coral polyps that do not have bilateral symmetry, but they do have symmetry. And then we have the bilateral symmetry where the left and the right side are the same.
Their closest relatives include simpler multicellular animals like cnidarians, such as sea anemones and jellyfish, stenophores, which are comb jellies, placozoans, and sponges. Analysis of Hox genes from these lineages reveal that the last common ancestor of flies and vertebrates likely had a cluster of about 7 distinct Hox genes, and these likely functioned in providing regional identity to cells along the anterior to posterior axis, as we see in modern-day flies and vertebrates. Looking further back gets complicated, as it's tricky to discern and interpret sequence similarities over such large evolutionary distances. However, it's clear that cnidarians do also contain a number of Hox genes in their genomes too. So the last common ancestor here on the tree, before the cnidarians and the bilaterian animal lineages split, likely also had at least 3 or 4 different types of Hox genes, though their exact functions are less understood. By contrast to the cnidarians, the known genomes of sponges contain a variety of homeobox genes, but no clear Hox genes. Even more distantly related fungi and plants also contain homeobox genes, but not Hox genes. It's therefore likely that the Hox genes arose early on in the evolution of multicellular animals, sometime after the sponge lineage had already diverged away from the other multicellular animals. Overall then, we can gain insight into the evolution of gene families like the Hox genes by tracing their ancestry across evolutionary trees that include the species of interest as well as some of their nearest relatives at key positions on the evolutionary tree.
You're almost done. Now we're down to our last 3 questions. So I'm looking for you to tell me patterns that you see in the similarities between the Hox genes in humans, chordates, and arthropods, fruit flies. So look at these Hox genes here. Tell me any similarities you see between these Hox genes and the ones in humans. Okay, and then tell me any differences you see in these Hox genes between arthropods or fruit flies and humans. And how do the observations support the claim that humans and fruit flies share common ancestors? So look for between those two differences and similarities. You should be able to relate how it relates to a common ancestor based on what you just saw in the video, based on what you see in this table up here, which was from the video. You should be able to do that. That concludes this activity. Woo! It was a long one. If you have any further questions, please ask your science teacher.